The cephalopod family is unlike any other animal species, I think, on the planet. These animals are incredible and they're very cool, so they're inspiring a lot of chemistry in the lab. My name is Leila Deravi, and I'm an assistant professor of chemistry and chemical biology at Northeastern University.
So what is it that chemists are learning from these creatures? That's what we asked Leila when the C&EN posse was in Boston in August 2018. We had a chance to visit her lab while they were working on a project that they just published in March of this year. She and her team are doing some very colorful chemistry, and we were psyched to chat with her about it. Leila and her lab work with cephalopods, which include octopuses, squid, cuttlefish, and other tentacled creatures. You may already know some of their cool biology. I mean, many of them travel by jet propulsion, and they can expel ink to confuse their predators. The cephalopod list of mind-blowing features continues, but Leila and her lab study one trait in particular: adaptive camouflage. The animals quickly change or adapt the color of their skin to match their surroundings, a quality that is particularly helpful when the goal is to hide from predators.
So we're really interested in studying the fundamental mechanisms of camouflage and color change and how these animals can do it so quickly and so fast.
The research team is trying to understand how our tentacled buddies achieve this on a molecular level, and then ultimately apply what they learn for human use. What they learn could be applied to a number of technologies. For instance, adaptive camouflage clothing for soldiers.
And we're really interested in the context of building sort of consumer products. So things that we're talking about lately are things that can be applied to the body, so maybe like cosmetics or creams that can color change or something like that, or even stuff like textiles and apparel that can color change depending on different environmental stimuli.
But before we see color-changing cosmetics or clothing inspired by cephalopod chemistry, there are some big questions that need answers.
The system is super complex. There's a lot going on in these animals, so how do they work? And so from my point of view, I approached it from a chemistry point of view, which is, okay, what are the small molecules and proteins involved?
And what that means is finding out what pigments and proteins are in the cephalopods. To do so humanely, Leila and her lab first anesthetize a cephalopod, in this case a squid, before sacrificing it for research. They remove its opaque outer skin layer while keeping the skin below it, called the Chromatophore Layer, intact. The Chromatophore Layer contains all the interesting, colorful goodness that Leila and her team are trying to understand. Next, they cut this layer into small sections and digest the tissue to extract proteins and pigment molecules. They can then analyze those using techniques including microscopy and spectrophotometry to learn more about their properties. And they recently found a surprise in the Chromatophore Layer. Leila co-led a diverse team of researchers from multiple institutions alongside Marine Biological Laboratory senior scientist Roger Hanlon. The group showed that there are two different types of colorful chemistry at work in the Chromatophore Layer. Researchers knew about the inky, stretchy pigment sacs you see here, stretching and shrinking. Like ink blots or globs of paint, these sacs contain pigment molecules that reflect certain wavelengths of light while absorbing others. But that flashing, iridescent sheen? That's the work of protein-based nanostructures scattering light. After light hits these structures, the rays scatter and interfere with each other to produce this color. It's like how a thin layer of soap can give a shade-shifting flare to an otherwise colorless bubble. This is called structural color. Although researchers also knew cephalopods use protein for structural color, they thought all these structures lived elsewhere in the animal, not in the chromatophore layer. Leila and her colleagues identified these unexpected proteins, called reflectins, in March.
And so we're piecing together, so there's proteins involved, there's pigments involved, there's light refraction and scattering happening in these animals, and when you have the whole system kind of pieced together, they give rise to these like beautiful dynamic color changes, and so that's kind of where we're at now with the story.
This is a very exciting and significant discovery because it reveals the intricacies of molecular, cellular, and functional adaptation that have been shaped by the power of evolution. Very few systems are more important than those of the chromatophore that provide both visual communication and visual camouflage in the teeming underwater world.
Leila tells us this fundamental discovery about cephalopod biology pushes chemists one step closer to realizing the consumer applications we talked about earlier. Although it may be a little while before we see color-changing cosmetics on the shelves, you can rest assured that the Deravi group will have their fingers on the pulse of what that it color is.
As you can see, we are very much in tune with the current trend, millennial pink.
How soon are you going to work millennial pink into your vocabulary?
Well, you know, we're really on trend with the colors in this lab here. The word millennial pink. I hope I say it more often. I hope I actually say it in my talk tomorrow. I think that would be a big victory for myself personally, and for what we're trying to get at in the lab, which is, really staying in tune with the culture and the trends of today. No, millennial pink. That's amazing.